So welcome to today's lecture. I think Brooke had announced that I would be standing in for him today. The topic of this lecture and the next overall is 3D function prediction. I'm originally a chemist, and I'm especially interested in interactions with small molecules. So the 3D function prediction will mostly be limited to interaction between a small molecule and a protein. Because the function of a protein is often that it binds to something, and that you can try to use 3D information in order to find out what does this protein bind to. So this is just to give you an idea of the general topic of today and the next lecture. And in order to look can we predict there, we're going to look at how does a protein interact with a ligand. And the other thing is, how can we predict which ligand it interacts with? And this is called docking, because it's like, you know, in some space films or something docks to a spaceship, and here we say that the ligand docks to the protein. Okay. And since this is a very cozy kind of setup, feel free to ask any kind of question. You know, this is your chance to ask everything about biochemistry that you ever thought would be worthwhile asking. I guess I have material to cover not the full 90 minutes, probably a bit less.
When we're talking about docking, do you mean binding sites?
Yeah, binding sites. Okay, the central, let's step back a bit. When you have looked at predicting a function of a protein, did you cover that this semester in the other lectures? So what's the general principle? What would you say is the general principle you apply when you try to predict the function of a protein? Very broadly.
We were looking at homology between sequences first. And then the thing when, like, what's the background? The professor told us it was a matter of evolution in the end. Genes that were evolving from the same kind of genes had similar functions.
Right.
And sometimes they have also, they develop an extra capability or an extra function which can be used to reply the function they're doing or to make another one. Okay,
yeah, so the point of predicting function is very often you just look for something similar. You know, this is really what it boils down to. And then the detail and the tricky thing is in defining what is similar. Because that's where it becomes a bit fuzzy, and as you said, the evolution might evolve apart. Something might get an additional function, and then similar is not quite it, or you have to define, okay, where's the limit? When does it stop to be similar enough to be able to predict function? Okay, well. In this part, we're also going to talk about similarity a lot, and then it's also going to be like, okay, and then how do we define this stupid similarity? So the observation people have made, first by just gut feeling, is that similar proteins have similar interaction partners. And this is also a bit what you said, like you see that evolutionarily, and it doesn't make sense, because you know, if you have a protein that has a certain function, then it might change a little, and it might, for example, if it binds a certain ligand and does something to that ligand, it might change a little in some part of the binding site, so the specificity might become different. It binds something else better, but it still does the same kind of thing on that ligand. And therefore it does make sense that similar proteins bind similar ligands. And then of course we can use that in order to say, okay, this protein binds that ligand, so therefore this other protein, which is similar to the first one, might bind a ligand that is pretty similar to my first ligand. And basically, if that is your take-home message from the two lectures, that's already a good thing to have learned. I have to... where did my question mark go? Okay, I have to say that this is not always true. Sometimes, as you learn, the protein might change its function, so you're not completely sure. Okay, so why are we interested in that? Well, first thing is we're interested in function prediction, as I said. The other thing to keep in mind, we're not going to talk about this a lot in this lecture, but it's still an interesting fact, is that pharma companies are interested in that a lot, because you can use it for drug development. One thing is that pharma companies have started to not just look at one protein target at one time, but usually they look at a family of proteins at the same time. Because you can, since they tend to bind similar ligands, if you make like thousands of potential ligands, it's more efficient to screen them against the whole protein family of proteins than just against one. Because you might, another protein of the same family might also be a good drug target, and then you reuse your compound. The other thing is, this is really interesting in order to find out about side effects. Because if you have one ligand, you say, I want this to bind to target my one protein, I want to block this one function. You have to look at, okay, but these other proteins are similar, so my ligand might also bind to those, and then I'm going to cause this kind of side effect. And if you start looking at that, you can also start to optimize against it. So you can look, okay, this is the other protein I might hit, and now I can try to change my ligand such that it only hits the target I want to hit, but it does not hit the target I do not want to be hit. And the other thing is that, but this is still pretty new, pharma companies have started to try and turn this around, this effect with the side effects and the families. Traditionally, and it's still the fact for getting a drug approved, you usually have to say, okay, this is the one protein I'm targeting, this is the mechanism how my drug works, and I only attack one protein. But in principle, often your target acts in a whole pathway. So there are several things acting after each other. And instead of just targeting that one protein very specifically, you could also target several things of this pathway. That is possible. But then again, you have to look for the side effect. Right?
I didn't get this last part lecture. Trying to react against a spectrum of...
Right.
Okay.
Yeah. So people are starting to, but it's still in the beginning and still being explored. They're starting to say, okay, we don't have single actors. We always have a network of things that do something. And why should we just pick out one thing? And there's also there has been an experiment to take common drug targets, so things that are used for drugs, proteins, and make mouse knockouts. Do you know that term, a knockout mouse? No, okay. So what people can do is to make certain genes dysfunctional, and then have that animal be grown. So they take a cell and they change something about it, and then they put it back into the mouse, and it's grown, and then they look at, do I see an effect? So is the mouse still able to live? Does it have a disease, whatever? And so this is called a knockout mouse because the gene has been knocked out. And they did that for a lot of things that are drug targets, and then they looked, do I see an effect? And only in 10% of the cases did they really see an effect. And if you make a knockout, that means you've completely changed the function. So it's not there anymore. And that shows that often the organism has other ways around a certain pathway which still get you the same function. This doesn't mean that drugs don't work. I guess probably a lot of drugs work because you probably are very often attacking more of this whole pathway, not just the one thing you try to knock out. But this shows that there is a certain point to the idea to knock out more than, or to attack more than one thing. Okay, but this is a side story just to out of interest. I always put some papers, and we're not expecting you to read that, only if you should be completely interested by a topic. This is a point where you could read up about it. Okay, so what can we do in 3D function prediction? Well, one thing is we can, if we have a 3D structure, we can try to find out where in this whole thing is the binding site. This is not always known. And we can predict which kind of ligand can bind to that. Traditionally, producing a 3D structure of a protein, have you talked about in this lecture about what it takes to find the 3D structure? Have you talked about 3D structures of proteins at all? I don't. Have you been in the last lecture, by the way, like last semester? No.
I've been to
You have, okay. Okay, so at least some of you have heard about 3D structures. Okay, so I won't assume too much then and step back a little. Well, producing a 3D structure is not so easy. Traditionally, it was something like a PhD thesis. Isolating enough of one protein, analyzing that experimentally. Usually you do that by X-ray crystallography or NMR. That was something that took you 3 to 5 years to get structure of one protein. And in that case, usually you were taking a protein that was very well studied, and you usually had an idea what the function was, and you wouldn't need the 3D structure to find out what the function is. Nowadays, this whole process has become a bit more streamlined, and people can do things by high throughput. And sometimes you even solve a structure of a protein that has not been so well characterized. And therefore, then in those cases, it does make sense to kind of mine and find out where could the binding site be and what could the function be. Otherwise, looking at this thing in detail is mainly a way to find out the details of how the protein works. Okay. So what are we going to do in this lecture? I'm going to talk a bit about protein structure, because I thought you might not know, or not everybody might have heard it. I'm going to go in a bit more detail about binding sites. I'm going to talk a bit about physical chemistry, because the interactions are very important in order to understand what we're doing here. And then I'm going to talk about what do we do to analyze binding sites, and next lecture is going to be more about docking. Okay, protein structure. Maybe you can make this a bit more interactive. Can anybody guess what this is showing?
These are alpha helixes and beta strands.
Right,
no,
this is
I don't see beta strands.
This is alpha helix,
Yeah.
this is beta strand. Can we start from the first thing?
Okay.
What am I showing here? Can you guess what the balls are? Any guess? I mean, it's fine. You don't have to know it. Just guess.
Certain amino acid, is it?
So one color is always one amino acid.
Yeah.
Okay, and what are the balls?
The balls.
They're like those little.
Binding sites.
No. So this is, here I'm showing the atoms that make up one amino acid. So this is, however, I have to say that usually when we look at proteins, we only show the heavy atoms. Hydrogen is so small and there's so much of it and so uninteresting for how this thing works in detail that we usually just leave it out. So if a chemist draws something, for example, if we have a molecule that is CH3, CH2, CH3, 3, we very often destroy it like that. Because the hydrogens are non-interesting. Everything that is not drawn is by default a C in organic chemistry. This makes it easier. And in this case, when we look at structures, we say, okay, there might be some H's attached, but they don't really make a difference structurally. Okay, so what you're seeing here is just a chain of amino acids. And you see that basically the backbone goes through and is more or less constant and doesn't change depending on type. But the thing that extends here can be different. And this is called the side chain.
Oh yeah, the rest.
The rest, right. And this is what makes the amino acids different. So the backbone is always the same. No, doesn't really change. The side chain is what makes different amino acids behave differently. And this is also what you know, when you looked at homology and alignments and things like that, this is what the letter stands for. Okay, now these things usually don't exist in an elongated form, but they fold in 3D. And there are some typical patterns which you see a lot. And one pattern is called alpha helix, and the other pattern is called beta sheet. And then the last exam, there was a question, is an alpha helix a more localized pattern than a beta sheet? Would you be able to answer that? We were hoping it was an easy question, but we found out it wasn't. So it's probably not going to be in this exam, but just out of interest.
Do we look at how they are formed?
Yeah, right.
To
yeah.
determine that?
Well, I mean, I haven't told you enough probably to know. Basically, if you form an alpha helix, every residue interacts with the residue, for example, if you take that one, 4 amino acids behind and 4 after. So it's pretty local. Whereas in a beta sheet, the main interactions are across the sheet, which could be somewhere else in the protein. So you don't have and you also don't have a rigid pattern. Here you always know that it's +3 and -3. And here it can be anything. And you can have parallel sheets, so the interactions advance in the same way, or you can have anti-parallel sheets. Okay, but this, for the chemist, this is basically just something that makes up the structure. It's not interesting really, it's just that something that stabilizes all the interactions. In the end, the chemist is interested in only this binding site. Okay, but to understand what we're dealing with, we have these alpha helices, we have these beta sheets which make the core. They fold in a way, and then you have interactions between that. And for example, here you see beta sheet, here you see alpha helix. They fold somehow. This makes this thing stable. This is called primary structure, secondary structure, so tertiary structure, and then you can have several of the chains that fold together, and this is called quaternary structure. Okay, just so you have an idea of what we're talking about. Now, when we show this, you don't see any of these side chains. Again, this is a simplification to be able to just see how this whole thing arranges itself. When we are looking at interactions with ligands, we will start to look at the side chains, because the side chains are the thing that interacts mainly. And also some revision, these are the different amino acids. As I said, these are the side chains, and these are what make the things different. I don't think we will ever ask you in an exam to draw any of these. However, it does make sense to get a feeling of what can be different about those amino acids, because it helps you also understand how substitution matrices for alignments work, etc. Because usually you see that the probability of replacing one amino acid by another amino acid is a lot higher if they have if they have similar properties. So, for example, whether you have an aspartic acid or a glutamic acid can be pretty non-important as long as you keep the negative charge. In some contexts, it might even not be important whether you have a negative charge or a positive charge. If you just want to have an amino acid at the outside of the protein, so it has to interact with water, you want to have something that is charged or at least has an OH group or something like that, so it can make an interaction with the water. In that case, all of these are pretty interchangeable. And as I say, you don't have to know them all by heart, but it makes sense to have at least a feeling for what is different about the things.
It's hydrophilic, right?
Hydrophilic, right. Hydrophilic, hydrogen-loving, hydrophobic, water repellent. Okay? And I think I don't know what you do in your exercises, but if you deal with proteins a lot, then you start to just know without learning. So if somebody says aspartate, at some point you know, oh yes, that's negatively charged because it's an acid and it gets rid of its H. Okay. Now I showed the overall protein structure. I showed you what the proteins are made of. And this is a typical picture how we show a binding site. Suddenly you don't see all this alpha helix stuff, etc. anymore, but here you see really the side chains of the binding site. Often we show it with a surface, because in here we're kind of just showing the center of the atom and connecting that center of that atom with that center of that atom. But of course that's not how a molecule can work. And it's not how a molecule really looks like. In the end, you have the atom cores, and you have the electrons around them, and the electron shells somehow interact with other electron shells. And if you really want to have an imagination of what a binding site looks like, then these funny sticks don't give us a very good picture. But we say, okay, we try to make something that is like the surface of these electron shells. And then we try to, for example, color it by the charge. So red is usually negatively charged and blue positively. Okay? And this is to illustrate how different things can be that bind to the same binding site. In this case, these are all, or probably all, synthetic ligands, so things that people made in order to bind to that and probably be a drug or some kind of inhibitor. But just to give you an idea how diverse the thing can be that can bind to the same binding site. And if we look at, if you want to get a feeling how it interacts, it's sometimes good to not really look at it in 3D, but to make a 2D picture, and then to highlight the interactions. Here, for example, there would probably be a hydrogen bond between those, a hydrogen bond between those, and I'm going to go into more detail about what a hydrogen bond is.
Are these numbers charges?
The numbers are distances. Because chemists usually start to get a feeling for if the length is this, then the hydrogen bond is good or not good. If it's too far distant, you can't build a good bond. If it's too close, then it would clash.
These things on the side?
This means there is an amino acid here, but it doesn't make any specific interactions. It just has a surface, and that surface might be able to interact with, for example, here you've got something that is pretty hydrophobic. You see there is no blue and red. That means only C's and H's, so it's hydrophobic. And then around this you would expect to have other hydrophobic stuff, and this means basically hydrophobic stuff is around here. And this helps chemists understand how the interaction works, and it might also help to get ideas. If I want to improve this ligand, then I could add something. Can't pick anything here, I think. Well, here my ligand extends somewhere, it's got an O here, and this doesn't show any interaction to the protein. So you could think, is it good to have the O there, or should I have something else which can interact better? Things like that. This is the point of looking at that in detail. Okay, so now you've got a rough idea of what we're talking about, what it looks like. Let's get into a bit more detail. How can we define what a binding site is? There are different ways of saying what is this thing we're talking about. One thing to define a binding site is to say, okay, we look at the function. Function of a binding site is to bind something. Now there are different things it can bind. It can bind other proteins, which is not the topic here, but there is also prediction methods for protein-protein binding. It can bind to substrates. That means this thing is an enzyme and it does something to the thing it binds to. So it catalyzes a reaction. Enzyme always is responsible for catalyzing a reaction. Do you all know what a catalyst is? I mean, what does it do and what does it not do?
It helps to make the
process faster.
Right, yeah, and that's a very important thing. It just helps to make it faster. It doesn't, so no reaction that would not take place without the enzyme will take place. It doesn't in any way shift the equilibrium, it just makes it faster. And it does that by lowering the energy to get the reaction done.
But since we have different enzymes, or in this case, are we considering only catalysts, or?
Yeah, different enzymes catalyze different reactions.
Okay.
And the thing, how enzymes can be different is one thing that can catalyze a completely different reaction. Like, for example, you can have a bond. This is called an ester bond, and this, for example, might be broken here. You could have a peptide bond, and this could be broken. And the enzyme might, in these two cases, it's pretty similar what you need in order to break that. But depending on the function that the enzyme has, it might need different things to happen around here in order to catalyze that reaction. The other thing is, so that's the reaction the enzyme catalyzes. The other thing is that around here, your substrate is going to have something. And this something which is around here can be different. Then the enzyme needs to bind those parts. And different types of the enzyme will look different in their binding site in order to accommodate different parts of this thing. And that means the specificity of the enzyme is different. So if you want to predict a function, it's interesting to find out what kind of reaction does it do, and what kind of thing does it do the reaction. These are two different parts of predicting something. Usually the catalytic site, so the things that do something here, is extremely well conserved. If nature has once optimized a mechanism to get this thing done, it's going to keep it in that way. And what diverges about a family of enzymes is the specific binding site for the substrate. So for example, what we need a lot in the body is something to cut proteins and cut peptides. And then you want this enzyme to just act on one type of peptide. And then you have different sizes of sub-pockets in order to bind only this or only that. But there are also some promiscuous ones. If you, for example, if you have something in your gut to cut what you eat, then you want that to be rather promiscuous. You don't need something specific for every kind of protein you've been eating.
Or if you want to help grow your antibodies, like make more, so you target some proteins that have enzymes that could catalyze or reproduce. Or
yeah, well, for antibodies, this is a completely different story. But Another thing is, for example, thrombin, which is responsible for blood clotting, needs to be activated. And you want this activation to be pretty specific, so you have a pretty specific activation enzyme there. That's in your blood, so that's the difference. And actually, there's trypsin, which is in your gut and cuts your food, and this is rather promiscuous. And there's thrombin, which is in your blood for the blood clotting. The reaction they do is, in both cases, they cut the peptide bond. But the specificity is different. And actually, these two enzymes are an example of things that are evolutionarily related. The sequence is pretty, you can recognize that the sequence is similar, but in the details they're different. Okay, so one function is binding substrates, and then it's an enzyme. Another function that a binding site can have biologically is to bind a cofactor. So in the example of heme, which is in your blood to carry the oxygen, you need to bind an iron atom, because this iron atom, hemoglobin binds the heme, and the heme is responsible for binding the iron atom, and the iron atom in the end is the thing your oxygen attaches to. So hemoglobin needs a binding site for the heme, and one thing you can try to do when you predict functions is to try to find out, okay, what kind of cofactor does this thing bind? Okay, so another definition for a binding site is just by form. Usually, if you have a cavity in the protein, then it binds it there for binding something, because why else should you have a cavity there? And on the other hand, can you think of why is a binding site usually a cavity? Yes. Yeah, just, you know, intuitively, but it makes sense to say it once. Yeah, I mean, so to put it a bit more scientifically, what do you do by having a cavity instead of just a surface?
Like this, you can only attach at this point. Instead, if it's like this, you can attach on multiple points.
There's one thing, you have multiple points to attach to, but there's also another thing. The degrees of freedom you have, like the movement you have, it's more contained. Okay, the last thing, which is pretty important if you want to program something, is you can define the binding site just by how can I find it as a computer program. And there we can use something, a convention about how we note down a protein structure. So there's a database called PDB, Protein Database, which collects the 3D structures. And the convention is that any atom that belongs to an amino acid gets the term atom. And then you see the coordinates and more specifics about it. Anything that does not belong to an amino acid is usually called heteroatom, het atom.
Can you repeat it?
Heteroatom, and it's abbreviated with het atom. So anything that is a het atom usually is something that binds to your protein, and then you can just look for proximity around that, and you're going to get a binding site. There are some problems to that, because sometimes people who try to determine a structure have to put in methods which help by finding out the X-ray crystallography details, and then it's a non-natural ligand, and you're finding a binding site for something that is completely artificial. But still, it's worth remembering, if you ever want to find binding sites, looking for het atoms and everything around that is a good first start. Okay, so yeah, I talked about that binding sites are usually a cleft in the protein. They are usually less hydrophobic than the normal interior of a protein, because usually a binding site is filled with water if no ligand is in there. Therefore, it needs to be more hydrophilic. And the form of a binding site is usually, or it's specific for binding something by its form, by the electrostatic interactions, by the hydrogen bonds it makes, and by the hydrophobic interactions. We're going to go into more detail on that. So what can you do if you're given the protein database and asked, please extract all binding sites for me? And we also want to have not only those that are filled, where you have a hat atom and you can look around, but we also want to have those that could have a ligand and we just don't see it in the structure. What could you do? How could you write a program for that? Well, you can use what I just said. It's usually a cavity, and I'm going to look for cavities. People have tested the hypothesis that it's the largest cavity, and indeed they looked in active sites of enzymes, and in 83% of the cases the largest cavity in the protein was actually also the thing they were interested in. Probably the other cavities also have some kind of function, but that's not the one that we're interested in. But so it's a good guess. Look for the largest cavity and you're probably going to have something that is relevant. So the first, meanwhile there are loads of algorithms to do that. And I'm going to show you two, because it's nice to see how the second one developed from the first one. It tells you something about how scientists improve their methods. And the second one is still in use today. But I don't mean to imply that this is better than any of the others. In the end, it's just, and what the point we want to learn you here is just how to think about this, how to solve a problem. Yeah. And a typical, I don't know whether Burkhard is doing this with you, a good way of revising, I think, is to think about, if I was an examiner, what would I ask about this lecture? And yeah, so I think something I might ask about this whole lecture would be devise an algorithm to find a binding site or something like that. And then I would like to see the important points. Okay, a binding site is the largest pocket, therefore I have to look for the largest pocket, and this is a way I could do that. Okay. So what they did, and this is pretty early, you can see here it's from '92. Computers were still pretty slow then. Therefore, they had to save time, computation time, and they took a very crude grid. Now you probably don't have any idea at all whether 3 angstrom is a lot or not. Do you all know what an angstrom is? Any idea? Okay, 3 angstrom, as you can guess, if it's a grid spacing, is a distance measure. It's and it stands for 10 to the minus 10 meters.
Is it the smallest distance possible?
Well, I mean, how can you have the smallest distance possible?
Between atoms.
Yeah. So it's not, do you know the SI? This is the standard system of unities. Usually you're supposed to say things in SI units. And a meter is an SI unit, and for example a nanometer is an SI unit. And 1 nanometer is 10 to the minus 9 meters. Actually, we chemists prefer the 1 angstrom because a hydrogen-hydrogen bond is on the order of 1.3 something. So 1 is like a bit shorter than the closest atom distance. So yeah, if we talk angstrom, we talk about the distances we typically have in any kind of compound. And you might remember the number you had seen there on the interactions, the hydrogen bond distance, that was 3 point something. That was between O they had left out the H and then, for example, an N, and then this was 3 point something angstrom. So if you have a grid with 3 angstrom spacing, then you are in a pretty crude range. You don't get really details of interactions. Okay? So the atoms are often a bit closer spaced. Okay, what they do is they take the protein, fill it with a 3 angstrom grid. Of course in 3 dimensions, but it's hard to draw them, so I just showed the figure in 2 dimensions. Now they go through the whole grid and always mark the point whether it's in the solvent or within the protein. So you have a grid which has got labels, protein or solvent. And a grid point is called protein if around it in 3 angstrom there is you find an atom. Okay. Now the next thing is to go along the grid and you mark every solvent point where you have found a protein before and a protein after. So you're not interested in, I have to get new batteries for this thing. You're not interested in solvent points around here because they're just solvent, but you're interested in those because you can go from protein to solvent to protein. That means it's between something. And you do that in all 3 dimensions. And some of these points might get labeled more than once. They might get labeled in one direction and also in another direction. And in the end, you look for clusters. And if you have that, you call these points pockets, and then you look for clusters of points that are labeled pocket, and the largest cluster is your binding site. Now the question is, how can you improve that? What could be a problem of this thing, and how can you improve that?
Maybe the spacing is, as you mentioned.
Yeah, spacing is pretty crude. That's one thing you can do. Can you think of any other things?
Could it be like a hole?
Yeah, you would, if there was a hole in the protein, you would also find it, that's true. That would be actually a really interesting thing to find because you usually don't have it. You would say, wow, I found a hole in the protein. Well, for example, if you turn this differently, it could be that you miss some of these things because depending on how the protein lies in your grid, you might not really go from protein to solvent to protein. And if you have more directions to go through, so the people who made Lixide, they also added the diagonals to make sure that you would catch something like that better.
The other one was only horizontal and vertical? Yeah,
well, 3 dimensions of course, but the third one hasn't been drawn. Yeah, but it was only horizontal, vertical, and the 3rd dimension. Yeah, and there they added the cubic diagonals to just get more. Another thing they added was they involved some computer scientists who helped them to be more efficient in searching for neighbor atoms and to say, okay, you can throw out this, you don't need to look at that part. Yeah, and the other thing is they now, because with the cubic diagonals, they have even more directions, and it makes even more sense to look at how buried this thing is. And therefore they label the points with the buriedness. How often do I find it? In how many directions is it buried?
What is buried?
Well, being between protein. You know, if it's like this, it's pretty buried.
Okay. So around it, let's say.
Yeah, surrounded. Yeah. And then you can kind of adjust how buried protein or how buried pockets you want to see, and how much you want to. And then you can say, for example, if I want to cut out something from my, later I might want to do an analysis with the binding site I found. And if I want to cut out the atoms and the residues that belong to the binding site, I just want to cut out those that belong to points that are pretty buried, or I just also want those that are more outside.
So all this research are done graphically?
Well, I mean, it's usually...
numbers in this thing? How do you say something is buried or not buried? Just looking at the interactions.
Yeah, usually for biochemists and bioinformaticians, it's an iterative thing. You know, you do something, develop an algorithm, then you look at the results. And actually, that's something we're trying to really teach our students, because very often, you know, nowadays we've got so many algorithms, people start throwing stuff at them, get out numbers, and be finished. And we try to teach that, you know, it's worthwhile looking at the details. At least take out a few examples, a handful or something, look at the details and see whether it makes sense. And then, in these cases, it makes sense to, or you should look that it does make sense in 3D. The stuff that you calculated has a meaning. Yeah, okay, so we had all these points. Now, the other thing we can do, now we just look at the geometry. The other thing we can do is to look at the interactions, the energies. Because in the end, the protein has to really interact with the ligand. And what we can try to do is find locations within this binding site where we think the protein might have favorable interactions with the ligand. And that's why I thought we need to look a bit about that physical chemistry to understand what kind of interaction energies we can have there. So what are typical interactions we find? Something you talk about a lot in biochemistry is the hydrogen bond. The way it works is that you have a water molecule, for example, a water molecule, or at least in general, you have an oxygen where the hydrogen is bound to. And this thing kind of draws the electrons to itself and leaves the hydrogen a bit more, the core a bit more exposed. And now this thing wants to still have a bit more electrons around it. And it starts sharing electrons with another oxygen of another molecule. So this is one molecule, this is another molecule. And this hydrogen starts sharing those electrons. And the electrons around this oxygen are not distributed completely uniformly, but they have a certain probability to be in certain parts of space. It's what we usually call orbitals in chemistry. And because the probability to find them in a certain part in space is higher, this also makes a certain geometry for the hydrogen bond. And now if you want to predict the interactions, you need to remember the geometry around an oxygen that is likely. Therefore, it's worthwhile to look at all this detail. Another interaction you see rather often is an ionic interaction between charged parts, and we also call these salt bridges. So, do you know what a salt is, by the way? What's a salt in general? You probably all know the salt you put in your whatever. Well, what is it chemically?
Sodium.
Sorry?
Sodium. Sodium.
Yeah, sodium and what else? So the salt you put on your bread that's sodium, and the other part is, anyone?
C. I would put the C.
Well, that's almost right. You also need the L. It's sodium chloride. But for a chemist, this is just one very specific example of a salt. The general thing of a salt is that you have two charged things. that form a lattice. Okay, and usually the salts are especially interesting in inorganic chemistry, because you've got loads of different kinds of atoms that can form loads of different kinds of salts. In organic chemistry and biochemistry, it's all organic chemistry. Salts are usually not so interesting, but as chemists we make the analogy that anything that is charged and interacts with something else charged, we kind of call a salt. Therefore, this thing is called a salt bridge. Just if you ever come across the term that you can remember, why is this thing called a salt bridge? Basically, it's just an ionic interaction, and you have these two charged things that interact somehow. And in organic chemistry, something charged is usually an acid, where the hydrogen has been taken off, or a base where you have an additional proton attached. That's the usual kind of ion we have. Okay, another thing is metal complexes. I won't go into detail about that because they're not extremely relevant. Something I learned when I prepared this lecture is that there is an interaction which hadn't been taught when I was studying. It's called cation-pi interaction. And this is when you have an aromatic ring. You know, so you can sometimes have molecules where the electron density is more or less smeared across the whole molecule. This is called aromatic. And when you have an aromatic ring, you can sometimes have a cation interact with that. Because again, you've got this charged thing here, you've got the electron density here above the ring, and they can make a pretty tight interaction in about the same order of magnitude as a hydrogen bond. So they are indeed relevant. And now if you want to predict how things interact, of course your program has to look for that. So it has to look for, do I find something that has an aromatic ring, and can I put something charged on top of that? Okay. The other thing we need to look at in order to understand how things interact is what happens chemically. The way we describe that is we have a protein, we have a ligand, and they form a complex. And always remember, this thing is always also working in the opposite direction. So things that can get together can also go apart. And interestingly, enzymes can also, of course, catalyze both reactions. Okay, and the way when we talk about interactions, and how tight an interaction is, we often cite this dissociation constant, where we take the concentration. So we look at the equilibrium, we say what is the concentration of the unbound protein, concentration of the unbound ligand, and the concentration of the complex. And if about half your protein is unbound and half your protein is bound, your dissociation constant is in the order of 1. Okay? And then if a lot of your protein is bound, then your dissociation constant is significantly below 1. Okay? So if you have a strong binder, you want to have a very low dissociation constant. This whole thing is driven by energy, and you can find a correspondence between the energy that is released by a reaction and the dissociation constant, or the reaction constant. And this is how you describe that relation. There are some constants, some natural constants. It depends on the temperature. But there is a direct association between the strength of binding, which you can measure, and the energy that is associated with that. And therefore, often if we want to calculate how strong something binds, we calculate the energy that will predict how it will bind.
Excuse me.
Yeah?
Could you read again the formula? The second
so yeah, this R is a natural constant, which I'm not going to go into any detail. It's just something that gives you the right scale, and T is the temperature. So this is interesting to keep in mind, that depending on temperature, it can be the energy that is released can be different. And this is your reaction constant. Something else to keep in mind, that this energy here is the so-called free energy. Because when something happens in nature, I don't know how much thermodynamics you had in any part of your life. The thing to keep in mind that is that you always have a part of energy that is released in form of heat, for example. But you also always have a form that is in the entropy. Entropy is a measure for, often it's a measure for disorder. If you look at quantum terms, it's a measure for how many states, in how many ways can you achieve that state. That's how it's related to information theory and things like that. And so a reaction can be driven by energy release, but it can also be driven by entropy gain. And if you look at normal temperature, room temperature, and you calculate everything together and go from natural logarithm to decadic logarithm, you can say that you have 5.7 kilojoule per mole times the decadic logarithm of K, and then you get an estimate for the energy involved in a reaction. So if you want to drive something, you can get an idea of how much energy does need to be released to get, for example, a factor 10 here in the K. Okay? Now, the other thing is we want to get an idea of how strong are interactions in nature. And in the gas phase, this is pretty easy to measure. Unfortunately, proteins are not in the gas phase, but in the aqueous phase, where it's harder to measure. But just to give you an idea, here we have been talking about a factor of 10 for about 5.7 kilojoule per mole. And if you have two ethane molecules, methane molecules interacting in the gas phase, that's about 2 kilojoule per mole. A hydrogen bond in the gas phase, it's about -22. But you have to say that in the gas phase, you don't have the water that's shielding or the electrostatics, etc. So this is an overestimate for anything that has electrostatic part. But just to give you an idea for the order.
And the last one is a salt.
Yeah, that's a salt bridge.
Yeah.
But that is really extreme, because in water that would be completely shielded because of the dielectric constant of water.
So in this case, the energy is low, right? So
yeah, so usually if something, so if, that's a convention of chemists. So if a reaction occurs by itself, you have a release of energy and you have a negative, and that has a minus sign. Okay? Okay, so if we want to calculate energies, we have to look at how do they depend on certain parameters. When we have to look at the physics. So, for example, we have the Coulomb potential, which is responsible for ionic interactions, and it depends on the charge and the distance and some natural constants. And especially this constant is important because in water it's different from in vacuum. So this is what is responsible for shielding. But we can calculate that pretty easily. We know all the physics for that. Unfortunately, physics is really good at dealing with single points and single electrons and things like that. But when you have whole molecules interacting, it becomes a lot more difficult. And here we start getting into an area where we start to approximate things. Instead of really looking at Coulomb interactions, we say, okay, we have Then we have two atoms. The atoms are surrounded by the electron shells. So the nucleus is going to interact, and the shells are going to interact. And we will have an attractive term because one nucleus attracts the electrons from the other atom. But we also have repulsive terms because the electron shells repel each other. And people called Leonard and Jones found that this thing can approximate the energies that we really see there pretty well. And yeah, this is what you get from that. And you've got the repulsive term, the attractive term, and together this pretty well approximates what you really see.
Yeah, and so here, so if the length is smaller, that means that there is higher energy required to form
to get the things closer. Yeah, so basically this is most relevant if you try to calculate the energy in hydrophobic interactions by just the shells approaching. So there is a certain distance where you shouldn't go further because it's going to repel. There's a certain distance where it's still attractive because one nucleus attracts the electrons from the other one. Okay. And the final thing we need to look at, and this is really tricky, is to try and look for the entropy. I'm sorry, I have this sort of really nice book, but I only have it in German. But I think there's meanwhile an English translation. But the point that it tries to make, you can still see, if you have a ligand in solution, then it's surrounded by water molecules which are bound to it, and which are therefore not free to go somewhere. So, but it is free to rotate. Now, if this thing binds to a protein, the water molecules are freed. Also, the water molecules inside the binding site are freed, so they're more free to move. You gain entropy. But on the other hand, this thing loses the freedom of moving around, and it loses the freedom of rotation, and therefore you lose entropy. And if you study it, so people have studied this in a lot of detail, etc. Very often, all these terms kind of cancel out.
But it loses entropy only in a local sense, right? Because in the global sense, molecules.
Well,
I mean, globally, like from the attachment to the.
Yeah, but that, I mean, it's fixed here, so it loses entropy.
Yeah, but I was thinking right now that it's attached, then they can move together.
Yeah, together they can move, of course. Yeah, but so there still, if you think of entropy in ways to achieve the same state, there's still less because they can only move together. Otherwise, they could move independently, and there's more. Yeah. And my point is, this is the part which we haven't really solved really well. This is an interesting thing, and it's pretty hard to predict how much entropy is going to contribute to ligand binding. But it does mean if you try to create a ligand that binds very tightly, and the ligand is rigid in the first place, so it's rigid anyway, then it's not going to lose entropy by binding, and that's probably going to make it bind better. Okay, let's make a short break. Or no, until when do you have?
Lecture.
Yeah, I mean, how much time do you have? Burkhard says you often start later and end earlier. And I thought this was driven by the time you have for the lecture, so it doesn't make sense to talk if you don't have time anymore.
No.
2:30.
Okay, good. Then I'll go on. Okay, so now if you want to find binding sites by looking at the energy, what you can try and do is find locations within the binding site where you think something could bind, where an energy would be favorable. And again, the first tool for doing that, do you have that? No, I don't have this citation here. It's pretty old. Grid is also from the 70s, I think. But still, this influences people and how they think, and it's kind of going around in the literature still. And what it does is to have different kinds of probe molecules, like you have a CH4, which is representative for anything that makes a hydrophobic interaction. You've got an OH probe molecule, which is representative for anything that can make a hydrogen bond. And you just go in a grid through the protein and calculate potential interaction energies. And the terms it uses is Lennard-Jones and electrostatic energies. And it also has a special term for the hydrogen bond, because as I said, this needs to be directional. Only in certain directions around an OH can you make a hydrogen bond. Yeah. Yeah, 85, so pretty early. And what this looks like is, here you have an example where you see where some ligands really bind in reality. You can take them out and then place the probe molecules at different places. This is just to illustrate that. Of course, you do that on the whole grid. And then you can make a contour surface of where you have a high interaction energy, or well, high and low is always difficult with negative terms. Where you have significant interaction energy. And then you can try and predict binding sites by saying, okay, I somehow cluster these points where you have significant interaction energy. And this thing is especially good for visual characterization. So when people in pharma companies try to make new drug molecules, they often look at how does one drug molecule bind and what other potential interaction sites can I see around there, and try to extend my ligand to also use those interactions. But of course, I can also try and use that for an algorithm. And that's not so early one. It's called Q-site finder. It takes just a very simple probe, just a methyl probe. That's just a CH3. And in that case, it has a 0.9 angstrom grid, so it's a lot better than this old pocket algorithm. And yeah, it's clusters adjacent grid points. And then the interesting thing, if you develop a new method, is that you always have to test it. Does it really work? And in order to do that, they took pockets where they knew where the ligand binds, extracted a certain distance around that, and then tried to find, okay, would I predict all the atoms that fall into what I really saw in reality? And they found that in 70% of the cases, their binding site overlapped with what they found in reality, and in 90% of the cases, it was among the first 3 predictions they had. And the overlap between their predicted binding site and the real binding site was about 68%. So that was okay. Now again, people tend to say, okay, this is a nice method, let's improve on that. So another group in 2008 said, okay, we can have a variation of that, and we call it I-site instead of Q-site. And what they changed is they don't just take a Cartesian grid, but they take the surface of the protein and then put dots around that in a method that is more close to where you would really find interaction partners. And the distance between the grid points is then a bit closer. It's 0.5 here, and it goes, when you go out, you go to 1 angstrom. And what else did they do? They also found that it helps to have 2 energy limits. So they have one energy limit where they have, so they go around, look for where do we find any kinds of points that are favorable for interactions. Then they have a pretty low energy limit for starting a cluster and a higher limit for extending that. Thereby they can get rid of things that are rather insignificant. And they also, instead of using this pretty crude grid force field, they use a more detailed one, which has got more terms for specific interactions. I told you, apart from hydrogen bonds, we also have these, for example, cation pi interactions, etc. And in this force field, a force field is always a term for a collection of equations to calculate the energy. That's what we call a force field. Okay, so they had a more detailed force field. Okay, so when we want to look at protein-ligand interactions, I think this is repeating something I had before. Then we look for all the things I talked about, how things can interact. And when we want to characterize a binding site, so now we come to the point of not just finding a binding site, but finding out, okay, what could it do? Then of course we have to start looking at how does it look like. One thing is, how does it look like in general, how big it is, what are the amino acids in there. Sometimes by just looking at the amino acid composition, I can also try to find similarities with others. I can look at how much water is in there, how hydrophobic it is, and what kind of electrostatic interactions can it make, and how can it interact with some other functional groups. Do you know, by the way, how you measure hydrophobicity?
You put water on?
Basically, yes, but a bit more complicated. So, I mean, of course, you can't take a protein binding site and put water on and find out where it's hydrophobic and hydrophilic. So you use indirection. One thing you do is you take organic molecules that have certain properties. For example, you take something that has just a long chain, so you have CH3, TH2, CH2, CH2, etc. You take another molecule which might have basically the same setup but an O here. And you take loads of those, which are always differ a little, and then you put them in a tube together with water and octanol. Octanol is it's got 8 C's, so 1, 2, 3, 4, 5, 6, 7, 8, and you've got an OH attached to that. That is octanol. So you can see you've got pretty much CH3 and TH2, and only a bit of OH. You put in the OH to make it possible for things like that to go in there at all. So you take a tube, you've got octanol and you've got water. You put in the thing you want to measure and you shake a lot. And then you look how much of my solute is in the water phase and how much is in the octanol phase. And then you make the ratio of that and you say that is the measure for how hydrophobic or hydrophilic is this thing, the probe I put in there. Then you measure that for loads of different compounds. And then you take that, you try to take apart this molecule and attribute the distribution you find to properties of the molecule. So you say the contribution of CO2, of CO, is so much for it to go into the water phase. And then you've got contributions for loads of these little things. And now if you have a molecule, you can go through and say, okay, I find this thing here, therefore it contributes that much to being hydrophobic or hydrophilic. Okay, and if you want to characterize a binding site, something that people often do is not really look at numbers, I mean like an overall summary, but to really look at the detail and map this to the surface.
The shape.
The shape, yeah, and yeah, look at it and see, okay, what's the impression I get? And the point is you always have to, if you compare two things, you may have to make sure that you work with the same set of parameters. So if you see a picture of one binding site in one paper and another binding site in another paper, you shouldn't try to compare that. Because they might have been using different parameters. Okay, you can also do something similar and map electrostatic potential to the surface. Again, it depends a lot on how you calculate the electrostatics, and it depends even more about your assumptions of which of the amino acids have the protein attached or not. Because the acids, the acidic side chains, might have got rid of their protein, then they're negatively charged, or they might still have it attached. And this again is a very tricky thing to calculate. But if you have different binding sites you want to compare, and you calculate that with the same program, then at least you get the trends right. So you can see, are they similar or not? That is something you can see. Another pretty neat thing that people have started doing is to kind of use big data. Instead of going to first principles and try to calculate everything by physics, they say, okay, we just look at all the binding sites we have. Where do we find what kind of interaction partner around which kind of interaction partner in the protein? Calculate a distribution from that. And then we map those kinds of distributions back to the protein we're interested in. And this is done in a program called Superstar. And this is also, like GRID, this is also used quite a lot by people who try to find out, okay, how can I make something that binds in there? Get your ideas. Where could a positive interaction be? Okay, so last few slides. If we now want to compare binding sites in 3D, the first thing we have to do is to align the structures. So find the orientation of one protein to the other protein that makes the right kind of residues comparable. And then we have to analyze how are they different, try to highlight for it, because the user will not be able to look at everything in detail. We have to highlight for him what is similar, what is dissimilar. And things we can look at overall is like, what is the pocket size? And are there some interaction partners there or not? And this alignment is pretty straightforward if you have sequence similarity, because making a sequence alignment is pretty easy. And if you have structural similarity, this is also still rather easy, because you can still find the alpha helices and the beta sheets and overlay them. Sometimes it's interesting to overlay two binding sites that are not really evolutionarily similar, but have the same function. These are the gems you find in all this, because then you can predict something rather new based on structure. And I'm going to talk a bit about how we try to do that in the next lecture. Just for this more easier things, there's a tool called Relibase. I think it's not being actively developed anymore, but it still works. So if you ever have two binding sites you would try to, or different structures you would like to compare, there's a publicly available version, and you can just go in and say, okay, I've got this kind of protein, now show me all proteins that have a similar binding site, and it helps you analyze how are they different. And the good thing about it is it stores the binding sites from all the structures in the protein database. And I haven't put it on there. It has it makes sure that the atom types are correct, because the crystallographers who deposit the structures are not always chemists, and sometimes the details of how like the C is a C, but for analyzing something it helps to know whether this C is in a double bond to something or not, because it will behave differently, and they make sure that all this is pre-parsed and calculated. Okay. And the really interesting thing we want to find is binding sites that are not homologous but still similar, and an example would be binding sites that bind those two things. because you can see that this part is pretty much the same in those two molecules. That part here is different, and the molecules that bind that are completely independent. But if you can find that this is similar to that, you could try to predict that they're also going to bind something like that. Okay, and this is going to be next lecture.